Assalamu alaikum. This is Shivar from Qaid University. First of all, I would like to thank the organizing committee of ICBS for providing me an opportunity to share my data. So today I'm going to present about the role of mitochondrial calcium in platelet aggregation and pro-coagulant platelets formation through adrenergic signaling. Let's start with the brief introduction of the platelets. As you all know that platelets are small anucleated granulated cells with a discoid shape and about 2 to 4 micrometer in size. Physiologically, they are important in hemostasis, tissue repair, and inflammation. Along with that, they also play their part in diseases like atherosclerosis, coronary heart disease, and brain stroke. Coronary heart disease is known to be the topmost killer in Pakistan. So, the risk factors of coronary heart disease like diabetes, smoking, and hyperlipidemia overactivates the sympathetic nervous system, which increases the production of catecholamines in the blood, and these catecholamines will enhances the platelet activation through PI3 kinase pathway along with the secretion of ADP. This ADP increases the intracellular calcium, which leads to the integrin activation and eventually to the platelet activation. The intracellular calcium, which is increased or released due to the secretion of ADP, then goes into the mitochondria through a specialized protein known as MCU. The calcium inside the mitochondria is overloaded, which triggers another opening or another mega channel in the inner membrane of the mitochondria, and calcium is leaked into the cytosol. This calcium may contributes to the formation of procoagulant platelets or the aggregated platelets. So our aim was to explore the role of mitochondrial calcium in the formation of procoagulant platelets and the aggregated platelets. We have started from the measurement of platelet aggregation using a time lapse based spectrophotometric assay instead of using an expensive aggregometer, and our results showed that upon addition of the adrenaline there was a decrease in absorbance, which is indicating an increase in platelet aggregation. And when we have analyzed the data statistically, there was a significant difference between the two groups. To further explore the role of mitochondrial calcium, we have then blocked the mitochondrial calcium through its recently identified inhibitor, that is MCU-in. And strikingly, we have seen a decrease in platelet aggregation in the presence of the inhibitor. So when we have calculated the amplitude and slope from the aggregation curve, there was a significant decrease in the amplitude of MCU in treated platelets as compared to the control, and similarly there was a decreased trend of slope in the MCU in treated platelets. It is generally believed that the calcium which goes into the mitochondria activates the mitochondrial dehydrogenases of Krebs cycle for the proper functioning of the mitochondria. So we have measured the mitochondrial dehydrogenase activity by MTT assay. MTT is a yellow color salt which in the presence of the mitochondrial dehydrogenases is converted into the purple formazine crystals. And our data depicts that, and our data shows that upon treatment with the adrenaline, there was a significant increase in the production of the purple formazine crystals, which is showing that which is indicating that there might be an increased level of calcium inside the mitochondria. Furthermore, when we have blocked the mitochondrial calcium with MCU-in, we have strikingly seen a significant decrease in the production of purple formazine crystals, which is showing that MCU-in might blocks the mitochondrial calcium. Furthermore, to complement our previous results, we have then measured the mitochondrial membrane potential by a dye known as TMRM, which is a positively charged dye, and it enters into the negatively charged mitochondria, and it gives a high mean fluorescence intensity. But if mitochondria are overloaded with calcium, then TMRM would not be able to enter into the mitochondria and its mean fluorescence intensity would be low. Our data on flow cytometer shows that upon treatment with adrenaline, we have seen a backward shift of histogram in this group as compared to the control. And statistically, we have seen a significant decrease in the mean fluorescence intensity of adrenaline treated platelets as compared to the control. Furthermore, when we have blocked the mitochondrial calcium with MCU-in, we have seen an unexpected results of mitochondrial depolarization in the presence of the inhibitor, and we have to just dig it out what is happening here. In the next experiment, we have then checked the impact of cyclosporin A or CSA on adrenaline mediated platelets. CSA is actually the blocker of MPTP. When we have blocked the MPTP with CSA, we have not seen any difference between the two groups. It is because that CSA is already known to enhance the platelet aggregation. In the next experiment, we have then measured the phosphatidylserine externalization in adrenaline-mediated platelets by a protein known as annexin 5. Annexin 5 binds with the phosphatidylserine when it shifts from the inner leaflet of the plasma membrane to the outer leaflet by the activation of the scramblase enzyme. When we have measured the PS externalization on flow cytometer, our data shows that there was a significant increase in the percentage of the annexin 5 positive cells in the adrenaline treated group as compared to the control. When we have in the next experiment, when we have blocked the mitochondrial calcium with MCU-in, we have seen a significant decrease in the percentage of the annexin 5 positive cells. So, in summary, we conclude our results as that adrenaline increases the intracellular calcium, which goes into the mitochondria, and this mitochondrial calcium may take part in may contributes to the PS externalization or procoagulant platelets and in the platelet aggregation. But when we have blocked the mitochondrial calcium, the PS externalization and platelet aggregation was inhibited. Thank you.